Well, thanks very much. It's a real pleasure. I'm Bruce Hecht from Analog Devices. I had 2 minutes yesterday on the stage, but today actually it's my pleasure to be able to host this panel, which is the last panel of this session, and I'm very excited about it. There are many topics here that I think we're all going to gain, and questions will be asked and things to ponder. So I'd like to welcome everyone at this one, the models of science and innovation, very much to the point. And we'd like to start with Carl Bergstrom from the University of Washington. He's a professor in the Department of Biology at the University of Washington and uses mathematical, computational, and statistical models to understand information flow in biological and social systems. I think this is really well suited, following up on some of the topics that we just heard about today. So we're really excited, so please welcome Carl to begin.
All right, thanks a lot. So my presentation today is going to be a little bit different from most of what we've heard, because rather than trying to directly apply models to data, I'm just going to look at some models that we build sort of from the ground up to understand how different components of the sort of scholarly ecosystem interoperate. I do have to figure out how to move things forward. Do I have a little clicker here or something? There we go. Okay, so just wanted to start off before I run out of time at the end by thanking my collaborators, Jacob Foster at UCLA and Yang-Bo Song, now at the Hong Kong University of China. So they were involved in all of this. And what I want to do today is really talk about, start out by thinking about, well, what is it, as we try to talk about generating science policy, what is it that motivates scientists? And we have this kind of caricature, in our imagination, scientists are kind of working away in a lab or a study trying to uncover pure knowledge about the universe, and that's in the imagination. And reality is probably a lot more like my friend Andrew Reed here, who's just trying to get on TED. Philip Kitcher, so, another way to say this, maybe even more cynically, is that these days scientists do research to get grants rather than vice versa. Philip Kitcher has a very nice phrase for this. He says, most of the philosophy of science imagines that scientists are epistemically pure. They're just trying to understand how the world works. But in practice, we're all epistemically solid. We're forced to be by the system that we operate in. We're economic actors just like anybody else. And what our job is to sort of think about, well, how is it that the incentives that are created by the institutions in which we do our science lead us to any kind of scientific activity? Today I'll talk about how do they lead us to allocate research effort across problems. And so, one thing we might ask, this is sort of David Hall has proposed, the philosopher of science has proposed that, we've got all these selfish agents, and yet it all works out great, even though we're all just pursuing, promotion and tenure and grants and fame and glory and all of that. Somehow the allocation problems all get solved by some invisible hand of science that causes everything to work out beautifully. And so, one question is, well, that's, it's one thing to say it, it's another thing to try to see whether that's actually feasible, or were there any kind of, is there any underlying modeling reasons that this might be true, such as we might be able to derive in neoclassical economics. So what I want to do, everything I want to do today is really trying to build up sort of, from the ground up, can we generate a theoretical framework for talking about the science of science policy? And what I'm doing is really sort of very much in the spirit of what was called the new economics of science, which was a great name when they came up with it like 20 years ago, and a little bit less good now, because it's less new. It's like the way that in biology we like to think back to the good old days when everyone was using next generation sequencing. So anyway, this is kind of in that framework if you're familiar with it. And what I want to really do is just kind of zoom in and ask kind of one question as a starting place, which is, why do scientists leave their workaday research and chase after hot problems that arise? And so, hot problems are these things that are sort of agreed by the scientific community to be very important. A kind of old one, how does DNA encode amino acid sequence, or what's the etiological agent of AIDS? How do we explain the Lamb shift? More recently, is Zika causing microcephaly? And if so, how is it doing it? Right? So there are these problems that are sort of well-defined in scope. They're widely agreed in the scientific community to be important. They're more rewarding than the sort of workaday stuff that we're doing in our labs. But they're not, it's important for my modeling that they're not just a grind. It's not clear how to solve them. You need some kind of ingenuity. And they're often useful to society at large, but they need not be. And of course, they're going to pull you away from your everyday research. And so the idea with modeling this is to try to build, to view this as a sort of classic microeconomic model, and look at what I call the credit race game, which is a model of what it is that we as scientists do. So we imagine there are N scientists. Each scientist has an outside option. The regular thing that she works on most of the time, her lab is famous for it. No one's really going to come in and scoop the majority of her work. It's productive. She can get her grants funded and her work published in her society journal and so on. And then along comes this new hot focal problem, and she has to make a decision. Okay, do I leave my regular work? Do I put that aside and work on this focal problem, or do I stick with my regular work? And so as we go and we look at each of these scientists, and they have to decide what are they going to do. If they do choose to work on the focal problem, then there's a race, right? They all start in my model, they all start at some particular time when the problem becomes apparent. And then they race to solve it, and of course, we typically use the priority rule in science for reasons we could talk about. First one to solve it gets all the credit; everyone else gets zero. The more cynical version, of course, is the first one. The most famous person to solve it within a year gets all the credit, and everyone else gets zero. But we'll go with this for simplicity. And so in my model, these are economic actors. They're trying to maximize the credit they receive. They vary in the amount of what I call scientific capital that they can bring to bear on the problem. Scientific capital is whatever it takes to get results. So in mathematics, it's pure brains. In molecular biology, it's pure money. And in most fields, it's somewhere in between. So they vary in this. And then they've got two choices. They can chase after this valuable focal problem, or they've got this outside option that will pay off in credit at some rate. So to enter in the focal problem, they've got to shift their research a little bit. They pay a startup cost in time and money to get going there. And they have this sort of constant instantaneous probability of solving the problem that's proportional to their scientific capital and inversely proportional to the difficulty of the problem. And so, of course, that means that the time to solution of the problem is exponential. And so we can write this all down. The probability that any scientist is the first to solve the problem is just what fraction of the total scientific capital working on the problem does that scientist represent. And so the solver gets some big payoff and everyone else gets zero. And so from this, we can write down the utility function or the expected payoff for the scientists of actually pursuing the problem. I'm not going to go through a lot of mathematical details. I've got a paper that I'll post to the archive in the next day or so that people can look at if they're interested. And so we start out by just looking for Nash equilibria of this credit race game. And so what's a Nash equilibrium in a game like this? Well, it's a set of players such that all of those, it's a subset I of the scientists, such that if everyone in I is working on the problem and everyone not in I star is not working on the problem, then nobody benefits from switching, right? So it's just any allocation of people to the problem or not, such that no one benefits from switching. And so we can prove now a whole bunch of propositions, just kind of doing a standard game theoretic analysis. Most simply, there's always a Nash equilibrium in the game. So to give you an example, we could imagine people find these mysterious symbols in Paleolithic cave art, and they seem to be some kind of code, and now they're these paleolinguists, Alice and Bob and Carol, and they can drop everything and try to decode these symbols if they want. And they vary in capital. Alice is a fellow of the Royal Society and has capital 10. Bob's an assistant professor with capital 5, and Carol's a Miller fellow with capital 4. And I guess my table's got on the line. That's how these things go on PowerPoint. But then we can sort of write out what are the utilities of each player. If Alice and Bob enter and work on this problem, the expected payoff to Alice is 6, expected payoff to Bob is 1. Carol doesn't enter. If she did enter, her expected payoff would be negative. That is, she'd be better off going with the outside option. If Alice and Carol enter, then they have these expected payoffs. Bob would have a slightly negative payoff, so he doesn't enter. If Bob and Carol enter, then they both get positive expected payoffs. But now Alice would enter too, because she's got so much capital she could probably win, and so this one's not a Nash equilibrium. We've got two Nash equilibria there. So the problem is, with all these kinds of economic games, we get these multiple equilibria, and then, how are we going to get any kind of predictive value out of our model? Well, we need some method for equilibrium selection. And what we're going to use here is a concept known as risk dominance. So a risk dominant Nash equilibrium is the one with the largest basin of attraction. It's the one that most states are drawn to. You can also think about it as being the one where there's the where that's the most costly for someone to mistakenly deviate out of, to behave wrongly at that equilibrium. So that makes it very stable if it really hurts anybody to step out of it. And so we can show that a unique risk-dominant equilibrium always exists. And in fact, it's the equilibrium in which the individuals with the highest scientific capital pursue the problem and everyone else opts out. So that's the sort of stable equilibrium that we might expect to see, is when one of these really hot problems shows up, the people with the most capital go after it. Other people can't afford to join the race. So in this Paleolithic code example, our risk-dominant equilibrium is this one up here, where Alice and Bob enter, Carol stays out. You see Carol would pay very high costs if she tried to switch in. And we have this other Nash equilibrium, but it's not risk-dominant because there's a very low cost to Bob if he were to try to switch in from that place. So this is sort of our first finding from this kind of model. Top researchers are the ones chasing hot topics. And we can extend this in various ways, and we can look at what happens when scientists vary in the value of their outside options. So some scientists have good things to work on outside, some not so much. And you can do the analysis there. I'm going to skip that because I want to get on to the issue of publishing partial results, which is another aspect of our scientific institutions that differ from other systems that get modeled. So if we think about patent races in academia, you've got to actually come up with the patentable product before you can patent it. But if we think, or patent races in industry, in academia, we are kind of racing for the solution of a problem. And at any point, if we think we're going to get caught, we can just stop and publish it and take all the credit up to that point. We allow these kinds of partial result publications. And so that suggests a modification where the game now takes place in stages. There's sort of sequential stages. Scientists can publish when they complete any stage of the game, or they can wait until they've solved the entire problem. So, for example, the scientists might race. Here, the scientist in green gets to solve stage 1 first. She goes ahead and publishes her results. Now everyone immediately knows that, and so now everyone's kind of starting from the start of stage 2. Maybe the purple scientist gets to stage 2 first. He chooses not to publish in this case. And that means that the other scientists don't catch up immediately, but he's not going to automatically get credit for stage 2 if someone publishes that first. Maybe perhaps he gets to stage 3 and now he publishes the whole thing and gets credit for stages 2 and 3. So this is like this new kind of strategic thing that comes in, which is that we can kind of publish at any point as we go along. And one of the things we're very interested in is when do we see the kinds of behaviors that correspond to this open science norm, where people are actively publishing things as they discover them instead of holding them back as lab secrets. And so we're very interested in identifying what we call public sharing equilibria, where people publish things as soon as they find them. And so we can show in this case that there's a unique risk-dominant equilibrium of this multi-stage game as well, and it's a public sharing equilibrium, and it's the public sharing equilibrium where all the top scientists up to some cutoff at each stage publish. So it looks very much like the previous risk-dominant Nash equilibrium, but now we've added this detail. And in general, so when will people publish partial results? In general, when a stage is very valuable, or when it's very easy, when there are many competitors, or the competitors have really high scientific capitals, when a scientist has low scientific capital herself, then she's going to publish partial results, or when the opportunity cost, the sort of value of the outside option is low. That's when people will publish. So to give you a example, so another kind of example, you have an unknown disease hits, and there's a couple of problems to solve. What's causing the disease, and where in the environment is it coming from? And so there's a couple of teams, one from CDC, one from LSU, that are kind of in place to solve the mystery. And so we can assign parameters here, and we can find the public sharing equilibria. And in this case, there's only one public sharing equilibrium where CDC and LSU both attempt the first stage. Whoever solves it first publishes immediately. Regardless of who solves stage one, LSU drops. The fixed costs of working on that stage are too high, and CDC solves stage two alone and publishes immediately. And this turns out to be more efficient than what would happen if these researchers couldn't publish partial results, because in that case, LSU wouldn't even enter at all. So by allowing publication of partial results, now we get LSU involved in the first stage, and we have a faster expected time to completion, which brings us to this issue of social welfare. This is so we had what do scientists do? Why does it matter? In our model, right, these scientists are looking out for their own interest, and what we want to do is, well, how does that align with society's interests? And because we model these outside options for scientists, we can talk explicitly about whether what the scientists are doing, are the scientists selfishly choosing to put the right amount of effort into solving this hot societally relevant problem. And so we think about social welfare. Ordinary research is generating kind of this increasing flow of knowledge that generates value over time. And when scientists choose to work on a hot problem instead, they put their everyday research aside and forego this value. But then once they solve the hot problem, that generates this sort of quantum leap of value, and so on. So that's kind of how we think about that. And so we can write down social welfare functions, and we can prove that there's a unique social optimum in this system where the scientists with the highest capital work on the hot problem and the rest opt out of the system. So at the risk-dominant equilibrium, which is what the scientists would do, we have both the at that risk-dominant equilibrium, it could be that we have under-participation or over-participation by the scientists. There could be too many scientists opting in, or there could be too many scientists opting out. I have an example that goes through this where we actually have scientists opting out. I'm going to skip that for reasons of time, but just tell you sort of the punchline, which is that contrary to conventional wisdom, allowing partial progress sharing doesn't necessarily accelerate the rate at which problems are solved. So why? Well, because allowing partial progress sharing changes the incentives on entering in the first place, right? Some scientists, the best teams, may choose not to enter at all if it's a game where partial progress sharing is allowed, and they can kind of be nickeled and dimed. So, unlike the Soviet scientists memorialized here, we don't have Lenin telling us exactly what it is that we need to be working on for the benefit of society now. We're choosing as these selfish economic agents, and because of that, partial progress sharing can slow things down. Wouldn't have slowed down Lenin-directed Soviet science. Bunch of further directions we can go with this. Very interested in what happens when the problems are of unknown difficulty. I'm working a lot on reproducibility issues, how these incentives shape, how the institutions shape individual incentives in ways that contribute to the reproducibility crisis. And then as we try to take this ultimately into sort of policy directions, we can think about a principal framework of, how is it that, say a government agency could act to shift scientists' efforts toward socially optimal allocation by investing grant funding or increasing the prestige of an area through prizes or other things. And so you can kind of see the questions here. I'm running a little long, and so I'll wrap up there. And thank you very much for your time.
So perhaps if there's one or two questions. One or two questions for Carl. And yeah, if you would like to set up Robert, that would be great.
If you've compared the output of your model to data, and what kind of data?
Yeah, so we have yet to compare the output of the model to the data. I mean, I think this is really one of those sort of models where, there are many different aims for models, and at this stage, we're still at the position where the model is really a way to just see how pieces interact with one another, the same way that very basic models in microeconomics or physics or whatever else might be. As we move forward, obviously, if we want to get to the point that we can start, using this as a theoretical basis for policy recommendations, we've got to find a way to bring these models into that very dangerous contact with the real world.
Other questions? I had a question, Carl, for the type of data you would need to test out the models. Is that something that you feel is accessible, or what kind of data would be needed to explore it?
Yeah, that's a great question. So what kind of data would we need for this? I think we need to, I actually haven't thought as hard about this particular model as about the follow-up, which is about problems of unknown difficulty and when scientists drop out of problems. We don't have very good theory about why scientists give up on things, and we can develop that out of this framework. And so there, I do think we could get the data we wanted fairly easily, or sociologists of science could get that data fairly straightforwardly, simply by finding out in different laboratories, how long do people work on a problem before they, how many problems have you worked on for a month and then just bailed on? Or how many problems have you sunk $10,000 into and then bailed on? And so those kinds of data, I think, would be very useful.
And maybe a last question?
No, I really enjoyed the game theory model. Just a suggestion for a refinement. I wonder whether the notion of capital could be domain specific, so that rather than having a single H for capital that you can move easily, certain scientists can have, be more ambidextrous than others, and you might not have capital for what you do and capital for an outside.
Yeah, so that's a great question. So actually, I glossed. We do have that in the model. I glossed over that. We have this model where your relative value of your outside option varies. So in the simple model, it's just proportional to your capital. But then we vary that, and that'll end up doing exactly the same thing that you were talking about, because it modulates the facility with which a scientist can move into the new domain. And what you find there is that you have a little concavity condition that requires that the top scientists don't have spectacularly good outside options. And so long as they don't have these spectacularly good outside options, you see the same thing where your unique risk-dominant equilibrium has the top people opting in. But yeah, I mean, sure, if they've got something else ridiculously good to be doing, then they won't chase that.
So I know
Thanks.
Carl may have to catch the westbound flight, so if we take the time now to thank him, that would be great. Thank you.
Thanks. Thank you.
So we'd like to introduce next Robert Axtell, who is with George Mason University, and we'll be hearing about technological epochs, which I think should be epic and epochs. So thank you, Robert. Welcome.
Thank you for the introduction, and thank you, Kari, for the invitation to come and talk. I'm going to be presenting a very abstract model, but I have spent 15 years of my life at Brookings here in town, and so at the end I'll try to relate it back to policy, at least notionally. This is work that's funded by NSF SciSip, so we appreciate the funding from there. Okay, so we've heard about this already several times in the workshop in the last couple of days, the idea of the hockey stick. In this case, it's the hockey stick of growth for the world economy. And in particular, I've written here terminology from the great theorist of the Industrial Revolution, Bob Allen from Oxford. So in 1844, Engels wrote about the immiseration of the English working class. And very soon after that, basically at that instant in time, their fortunes took off. And basically the rough number here, though it's a little hard to read it in non-log coordinates, is that since the 17th century, we've had roughly 100-fold increase in in output per person in productivity. And so the quick calculation is that every year it's a rather modest return, but it's in essence a 1.3% growth in output over these time periods get you basically this 100-fold increase. So when you think about 100-fold increase, what it means that the work that would take us 2 weeks that would take our ancestors 2 weeks to do, 80 hours, we can do today in 1 hour. That's roughly the calibration. So an incredible increase in this technological capability. Now, what is the conventional rationale for this, or conventional explanation for it? So here, this is from Carlota Perez, her succinct summary of these 5 great tech epochs. And so even though this plot is semi-smooth and it shows this progressive increase in productivity over time, she will divide it up, and it's normal to do this, to break it up into these kind of ages of electricity and oil, and now we're living through the information age. Now, she believes that there's several reasons, several things that make an epoch, and these interconnectedness and capacity things are down below. I've just put in here conventional wisdom. What we'd like to do is have a model for this. How do these epochs arise? Now, of course, it's intimately tied up when you read the histories with the features of steel, with the fact that oil was found in western Pennsylvania. I mean, it's tied up with a lot of historical just so stories. But the question is, are there ways to make models which actually get to this idea of technical epochs? Just by way of background, one thing that I'm not going to focus on too much today is the space of all possible inventions, in the sense that this is from Shear's work on the value of inventions, and basically roughly 10% of inventions have about 90% of the value. That's the conventional wisdom here. Most innovations don't amount to much, in the same way that most papers don't get cited. So it's these heavy-tailed distributions have these curious properties. So in essence, we want to model basically processes by which innovation happens. Most innovation is done, it's commercialized by firms. And so just to give a sense of where the whole literature on firms has gone in the last couple of years, those of you who are not economists, I mean, these days we have the data on all firms in America. So 6 million firms, we have employees, 120 million workers. We know the gross empirical regularities associated with these firms, and we can today build models that move beyond the conventional approach, and we can try to understand what is the structure of these, in the case of the U.S. at least, which we have the data, case of the American firms. So what I'm going to try to, I will not talk about this book in particular, but I'm going to talk about, though, kind of how firms innovate, but with a very abstract model. And so here's how the model is going to go. Okay, we're going to model technology evolution and kind of accept Marburger's challenge, which says, how does it work? How does technology manifest itself? What makes it go faster or slower? And then I think the normative question, I'm using the word positive and normative here in the way an economist normally would. How can we accelerate it, say, for policy purposes? How can we accelerate the technology process? Now, to an economist, it takes a model to be a model. Now, I've heard today about narratives and about how congressmen don't want to hear about models and things. I mean, at least when it comes to, many of us are academics, you can't just beat a model with a narrative. You have to beat a model with a model. So I'm going to propose a model here. So my question is, what is a minimal specification sufficient to produce epochs? All right, so not continuous evolution, but epochs. All right, and then we're going to build on the ideas of technological recombination that's implicit in the theorists of technology change, Schumpeter, Brian Arthur, Weitzman most recently. So here's the basic idea. I'm going to apologize for the details here. I'm going to give a picture of it in a second. We're going to imagine we have a bunch of agents who can hold in their possession at most some number of goods at a particular time. At time 0, there's some seed set of goods. Imagine a small number of seed sets. A seed set, which is this seed set is larger than how many goods you can hold. So you can't hold the whole set yourself, but then the number of agents does in fact have the ability to hold all the goods, so they're not going to go extinct right away. Now, every good does have some meaningful fitness, a la biology, and this is a little bit non-economic, but imagine that good has some fitness. At time 0, the seed set is all seeded to have fitness between 0 and 1. That's going to evolve. Now, every agent, though, makes a subjective assessment of that fitness. So it basically draws a random number and says, "Now, this cell phone, it's not very useful. I don't want to have it." Okay, that's one possible way to assess a new good. So what agents do is they say, well, we're going to try to innovate, but we're also going to consider adopting the new goods that other people have innovated, and we're going to talk to friends about what goods are actually worthwhile, which ones are not. So innovation by recombination means we start out with this seed set. You pick two. If you're an innovator, you pick two goods at random with some small probability, cap P. You say, good K plus good M makes good Q. And then Q has fitness, which is this once again, just kind of random combination of the fitnesses of its components. So you imagine, an iPhone is made up out of out of a GPS system and a telephone. So the fitness of the iPhone is a combination of those two things in some way. It doesn't matter really, it turns out, what you put here for functional form, but you're going to get the same qualitative results that I'm going to show you in a minute, no matter what. So the average fitness is going to increase over time. The goods that get created can increase over time in their ability. We're going to then make this new good available for adoption. Okay, so now this idea of diffusion is, I'm not sure why I've lost the color here. Hopefully you can read that. Basically, once a good has been invented, we're going to basically offer it up for adoption to the various agents. They're going to basically just pass it around to their friends and say, do you want this good? It's a really good good. You should try it, that kind of thing. Okay, so here's the picture anyway. Here's the picture. You have an agent's carrying around N goods. Suddenly, there's a new one invented. Then it likes that, it keeps it, it has to throw away some other good. It then shows that good to a bunch of other agents in the economy, and that ends up being that a lot of them like it, and they end up throwing a bunch of stuff away that they didn't need anymore. Okay, that's all there is to it. So you get the sense that over time, what's going to happen is innovation is going to create new goods, stuff's going to go away, every good's going to have a finite lifetime. So I'm going to show you now a typical kind of qualitative what happens in this model. This just shows that, here's the seed set of goods, and you're going to get this evolution of new goods over time. By the way, those of you who have ever looked at the artificial life models, this is very much like an algorithmic chemistry model there. Okay, what happens in this model? The simple basic idea is that there's only 5 parameters: number of goods, number of agents, number of the probability of acceptance, how much diffusion, et cetera. And so we can exhaustively evaluate all these parameters, and I'm going to show you kind of a typical regime with 20,000 agents. You can only hold 10 goods. There's 20 in the seed set. And there's a very small probability of invention, et cetera. So here's basically some random time series for how many goods get invented. But this is a typical, after some long period of time, this is a typical lineage. We heard about scholarly lineages the other day. This is a lineage of some good here that got produced, but this is what's made out of those two, and that one's made out of those two. You can check all the way back to the seed set if you look all the way back. So every good has this lineage, and then it can be tracked here kind of with no noise. This just says every good only survives for a finite amount of time in the economy and it goes away. So after some long period of time, the goods are, you have some completely new set of goods you didn't have before. Now this is going to show, this is the number of goods in the economy, and it shows this kind of creative destruction out of the Schumpeter, right? Some good gets invented, and all of a sudden you wipe out half the goods in the whole economy. They were there before, you don't need them anymore. What was the third most popular, most common profession in 1900? Cooper. Making barrels. We don't need coopers anymore. This model produces then exponential growth at a certain rate, and you can tune the up growth to be large or small. But the welfare that agents get is heterogeneous here. You're seeing the kind of distribution of that. This is just saying that how many agents adopt the particular goods at a certain time, and you see this very skewed distribution, or very skewed representation. Now, but turns out, I said every good that comes along gets knocked out, right? So this is either a plot of how goods get knocked out, or it's a perfect picture of a city by a frozen lake. You can call it either one. So what you see is that the colors that go up at one point actually come down later on. So that good was heavily adopted, almost every population took it, and a little bit later, everybody got rid of it. Right? So this is perpetual turnover in the number of goods. Anyways, this is the emergence. This is the surprising result of this very simple model. We get the emergence of technological epochs here. So what each one of these quasi-battleship-looking configurations is, there's a sudden spurt in the good space up to something that's very fit. And then the only thing that's going to be adopted subsequently are things that are derived from that very fit good. So let's just blow this up a little bit and look at it. So this is over time. This is the kind of the picture of which goods are adopted. The colors don't mean much, but the sizes refer to how many people have adopted it. So I'll just point this out here. So here's some technological epoch going along here. Something gets invented, but it doesn't take off. It's very fit, but for whatever historical reason it doesn't go off, it doesn't take off. Here, this one does take off, and now it produces this whole new lineage of goods. And this just says that most of the people in the society are now using it. Well, a bunch of other invention happens here. A new epoch takes off, then this one just whittles away. Notice, it's also the case that stuff can even get reinvented. So this guy kind of invented, somebody actually reinvented the darn thing here. This keeps going on and on and on. But now remember, you're living in the world of red queens. And so if we actually, this is the absolute fitness. The relative fitness is this terrible thing, which basically is the same picture, but now the most fit thing that gets invented after a few periods is basically not fit anymore at all, and the next best thing comes along. Okay, so this is I say very simple model. To me, it has the surprising result that it produces these epochs, even though we haven't built in this idea. At least we don't think we have put that in explicitly. Okay, so the quick summary of this is that, and my time is running out, is that I've argued qualitatively that policy needs models and that models need data. Now, I haven't shown you so much data today, except to say that in the work that we've done on firms, we use all the firms' data. Because in the data we use on firms, it's based on IRS data. We only have one field in there that's actually related to technology, and that's going to be R&D investment. And that's a very noisy field. And I've written here that's this area that we've been working in the last couple of days. It needs models and it needs data. I think it's not sufficient to say it just needs data. It needs to have models also to inform what the data mean. Let me just mention by way of I guess advertising for a few months from now is that there is a National Academy report coming out called, with a terrible title, Modeling the World. So it's meant to survey all possible the full span of models that are used by academics to do various things, from climate models to social science models, et cetera. And I've been part of this committee, and say the report is largely done, and it will be released sometime this late summer, I believe. And I'll stop there. Thank you.
Thanks, Robert. I think maybe we should continue with the question. Thank you for sticking to time. I'll give you the first, and then.
So there are obvious connections to various work in macroevolution, and I'm curious if you've explored any of that at all, because there's a bunch of new theory being developed there, in particular tracing characters of different lineages as they advance and their propensities for further evolutionary change.
I guess the short answer, Carl, is not explicitly, although I have to say some of the Gould-Eldridge work was motivation for this, or kind of gave us some sense of where to go, but we've not docked with that literature at all, so we should do that. Yeah, sounds good.
I was being an old Schumpeterian at heart. One of the things I was noticing was the overlap of the battleships. When the new one comes in, it's actually after about 1/5 of the second one, it isn't delayed. So are there particular parameters that actually delay when you get the first signal of when the next revolution? Because most of the histories that I have seen, the next revolution is usually about 3/4 after the first battleship has gone by.
Yeah, about 50 years goes by or something, right?
Yeah, about 50 years.
Yeah, so it turns out, as you change the parameters, you can either separate the battleships in time or that way too. So you can shift them around.
But can you move when they overlap like this?
Yeah, so it's a good question. I think that
If that's too close.
there's a long paper on this. Yeah. There's a long paper on the model, and what I can do is I'll send you the paper. It goes through many more parameterizations and things. I don't believe there's not a plot that's going to satisfy you, but it may be that we can doctor the results, as it were, in order to get what you want. Yeah.
Right, Robert. First, I got a question on this. Was there a button that we pressed to get back?
I don't know.
Okay.
I just
Maybe Dan will help with that. All right. Well, my other question for you was going to be, you were describing that the epochs, come out of the model that you've shown. I was just wondering, is there a contrast with that and the idea of logarithmic, on a logarithmic scale, Moore's law, and other things that, sort of a technical,
That's right.
progress will be made
exactly.
somewhere. Do they fit together, or are they contrast one to the other?
It's a good question. Some of you may know this.
Dan, can you check?
There's been a bunch of recent work. I'll get the microphone. There's been a bunch of recent work on the relationship between Moore's law and then experience curves, which this is Wright's law, so-called, right? That you can build the next airframe for less than you built the last airframe, and then so-called Sahil's law. And those are all interrelated. Those are all exponential laws come power laws. And we've not related our work to that, but I think that's important to do. But I recommend anybody who wants to learn more about that, at the Santa Fe Institute, where I'm associated, there is a great website which has computes actually these Moore's law type things for 100 important technologies. And then I think the way that those are quite heterogeneous, quite variegated. But I think that it's a rich data environment to actually try to calibrate this kind of model too.
That sounds great. Well, thank you, Robert.
Thank you.
Thanks very much. Thanks again.
Okay.
So we'd like to welcome James Evans from the University of Chicago. He's here to talk to us today how science thinks and how to think better. I think we're all interested in science, and as we heard earlier, the humans and the machines, and where is this taking us? So please welcome James.
Thank you. I'm delighted to be here, and I'm only sorry that I didn't get in earlier. I'm going to reflect on a handful of different models connected with data, and really, I think I find policy insight about the first and the second question, how models of science and innovation improve scientific decision making, kind of comes out in some ways in the contrast of these models with one another. And I'll also talk about briefly at the end a simulation that kind of I think helps us think about the potential for some kinds of science policy decisions. So. So basically, I'm rendering or thinking of science as a complicated, complex system. It's got diverse, differentiated components. These are technologies, they're individuals. There's strong interactions between those components. There are emergent, often unexpected collective behaviors that take place at multiple scales. And like this Hadron Collider, right, which kind of sussed out the Higgs boson and another kind of set of unexpected bosons last December, like the exceptional group E8. This is a paper that talks about mathematics as a complex system, right, where there are enormous proofs, often computational agents, which are facilitating and participating in the context of the development of those proofs. And we have systems which are themselves, the attention ends up being an emergent property, like Carl was talking about earlier. These hot topics are something that are potentially unpredictable and emerge from these complex interactions. And I'm arguing that they do have a potential linkage to science and innovation policy. Now, my model of the human in these analyses is going to be relatively simple, but I'm going to argue that even like the ant, simple programs, simple interaction set, but then linked to a complex environment, you have the potential to create these really elaborate structures of quite marveling and staggering beauty, like the National Academies, where we are right now. Okay, so the roadmap. I'm going to talk about biomedical publications as a kind of a substrate for scientific thinking, for the community's thinking, how the community thinks through the possibilities. And it's going to reveal some heuristic reasoning pathways. I'm going to argue that the cleavage, essentially, of scientific thinking to that publication space ends up detaching it from other classes of spaces, like health problems in society, for example. And then I'm going to talk about setting up the basis for simulation against data that evaluates the way in which scientists select problems, kind of assuming now that this publication space reflects a really rich set of scientists' options and opportunities, and then we'll simulate a rich set of counterfactuals to leverage this for science and science policy. So this is our data, a stack of papers written by people. And this reflects a paper published last year with Jacob Foster and Bill Shee. So kind of you explode those papers, there's people and there's methods and there's subjects, in this case diseases, and there are kind of agents within those subsets. They're chemicals, which end up being traces of diseases and also in some cases traces of methods in this space. And they're all kind of connected in this enormous network. And so I'm going to argue that the scientists think through this complex network of context and content. And I'm taking as inspiration this idea from Alan Newell and Herbert Simon that there's this network of possible wanderings in a problem space, and that possible set of networks ends up being very meaningful as scientists drift through it and searching for all opportunities. So the data that I'm going to use here, again, is that stack of papers, but it's been curated and processed by an army of postdocs and research annotators at the National Library of Medicine. And we're going to extract from those 20 million authors, about 9 million, from those 20 million papers, about 9 million authors, about 9,000 chemicals, 4,500 diseases, and 2,500 methods from 1950 to the present. And we see that things are changing over time, right? So basically, the number of authors is increasing relative to the number of papers, right? That's not surprising. That the number of papers in which a method ends up being involved increases relative to the number of papers that any particular disease is involved, which is to say that molecular biology creates the standardization of labs, right? And so we have certain kinds of organization that are taking place over this time. So certain methods end up being enormously important over this period, while others remain quite sparse. And the papers themselves, as artifacts, end up being enriched over time. So they grow from one to two authors over this period. They go from 2 to 4 authors over this period, and basically the methods end up growing commensurately. So there's kind of a labor theory of value. There's more, even though science is easier, as Robert suggested earlier, it's like we require more of these artifacts to get published. There are more experiments, there are more people, there are more techniques, and there are often even more subjects. Now, if we think of this thing as an enormous networked space, is a hypergraph, right, where everything can connect to anything else through anything in this space. So I can connect to, my collaborator through a disease that we're studying, through a method that we're investigating. I can connect to a new chemical through a disease for which it's a trace, right? So if we think of that entire network space, everything's close to everything else. Both in terms of what ends up getting connected in the next period and just in general, right? At the state space, everything's close to everything in this very dense scientific system. And so we're going to render and model this as a hypergraph. And so basically, papers are like sets. They contain people and objects, diseases, chemicals, methods. And we're going to look at what that says about the history and evolving structure of science and the degree to which this literature reflects the opportunity space that scientists find for themselves. So this paper, NAP protects hippocampal neurons against multiple toxins. So, we can pull out these elements of the paper and render things like this. So a new paper ends up connecting these two therapeutics with drug-induced liver disease in the context of rats, right? And so in 1982, right? So everything's close to one another, but things get hyper-linked and networked in this space. So what we're going to do is we're just going to say, what if scientists just randomly walked along this space that's available to them at any moment, right? So they randomly walk along this side. In fact, if science is a representative agent, just randomly walks along this space, how much of next year's science does that predict, right? If people are just wandering locally. And so what this does that's different from like a nearest neighbor model or a standard link prediction model is if I'm trying to see the likelihood that I connect with another scientist, scientist A and scientist B, I could go through these many things, like, for example, a shared co-author or a disease that we both study or a chemical that we both use or a method that we both deploy. But if that method is enormously large, if there are many, many people looking at it, it's very unlikely I'm going to get to that person through that method, right? If I go to a small conference, I'm likely to meet you and get to know you. If I go to a 5,000 person conference, it's very unlikely that we're able to connect. And what happens is we're able to predict about 95% of the connections of diseases, chemicals, new co-authors, et cetera, in the next year as a function of random walks in last year's papers, right? So we don't have institutions, we don't have all these kind of social things that we typically, so this space, it's not that these are the only things that people are doing, but basically much of the serendipity that people might otherwise experience ends up being embedded in this scientific record and predicting the likelihood that scientists, if they were just randomly walking, end up showing up at the papers that show up in next year's journals, right? And we can go down further and we can find that, for example, one thing that I think is interesting for these models is that things combine with other things through other types of things. So authors connect with other authors through things other than authors typically. And diseases connect with methods not through diseases or methods, but through chemicals, right? So it turns out that adding more data in this case ends up bringing superlinear increases in your ability to predict next generation science. So it's not, so our findings suggest that just picking one trace ends up dramatically distorting the way in which you might view evolution and novelty and all these kinds of things in this case. And we can also basically show that the things that we predict that you walked along to get those people together end up collecting in the papers that those two authors wrote, or in the things that other, so empirically it looks like people are actually following these pathways. So in short, just, scientists appear to think through the literature by wondering. Locally, scientists associate between layers and bias against within layers, and partly that's because of the geometry of something like a paper. You can have just 15 methods and no people yet. That might be next generation papers. But then if scientists follow the literature, like if the literature accounts for so much, essentially, of next year's papers, what doesn't account for next year's papers? Well, if scientists follow the literature, what do they ignore? So these are, so combining this with another analysis, there are a handful of studies in which we analyzed and found that the consequence of prediction in the literature is that there's no relationship with the burden of disease, for example, in the biomedical literature, right? So the fact is, it's like, science is like Seinfeld, right? It's like it creates its own problems, these little microdynamics where you can just stand in line waiting to get in the restaurant for like 30 minutes, and that's a show. It's like, people are creating their own problems, and these things loom so large that they eclipse, essentially, in this particular case, the burden of disease, the pain and suffering that's distributed. Now, this is true at the level of the world. As you can see, this is the kind of the disjoint distribution of life year adjusted loss to disease and research articles or investments in the following years. And you could say, well, okay, for the world that makes sense, because there's like a different distribution for rich and poor countries, and it turns out that like rich countries spend way more in science, and so that kind of like makes sense, But it turns out that if you look just within the US, you just take the insurance records, right? You find the same thing. You find a disconnect between problems and disorders, both statically, so big problems don't line up with big research, and dynamically, growing problems don't line up with growing investment and vice versa. And so, for example, you have things like your breast cancer has enormous attention in the context of relative trials, clinical trials, and also relative research attention, but as a result, it kind of has an opportunity index, as we construct, that's actually really quite small relative to its burden of disease. And so we also can kind of construct these heat maps that kind of show light in the top on things that get more attention than they deserve relative to these burdens, and beneath those that they get less attention than they deserve. And then we kind of construct, opportunity indices and whatever. The whole point is that models and metrics, I think, can help us rewire scientific attention. Now, I'm not saying that we should just like shift all our money to like study the things that are like the biggest problems, because why aren't we studying those? Well, okay, maybe it's because we're like surrounded by delicious scientific internal problems. Maybe some of those problems are hard. And if we just like banged our head against the wall, we would get nowhere. But at the same time, I'm arguing that if we shine a light and bring in essentially these other pieces of information that get eclipsed essentially by an increasingly large and complex scientific system, then we can do other kinds of engineering. We can predict bottlenecks. We can predict things that would be relevant to essentially solving problems in a particular area. And which investments could essentially nudge research towards those things without sacrificing the quality and the momentum of research. I have just a couple of minutes left, and so I'm just going to whip through a final kind of example and a half. And that example is a couple of papers that came out with some colleagues of mine, Jacob Foster, Ian Foster, no relation, and Andrei Rozhetsky, in which kind of taking seriously this idea that published science ends up being a substrate for science, can we start looking at choices that scientists make as a function of this map and evaluating those choices. So initially, we did a study in which we were interested just in how scientists choose the next experiment. There's lots of things that could go into that choice. One of those things is following the crowd or kind of striking out on your own. And that choice ends up also being a choice between like ensured productivity versus taking a risk that you might succeed, that you might not succeed. And this essential tension ends up structuring all kinds of scientific activity, from publication to jobs to tenure, versus the best publication, awards, enormous outsized prizes. And so for this study, we just kind of rendered scientific choices as a network, and we kind of chopped that network up at its joints. And so we instituted things like, so jumps are when you bring a new chemical into this space. New things are where you take existing things and you connect them together. You could repeat things that occurred before. You could do things within existing subfields, or you could bridge between subfields. And I'm glad Carl's here because we used to build those subfields his map equation approach that essentially random walks through these spaces and builds addresses that are local to each other. And they end up producing very believable, very plausible scientific subfields. Thanks, Carl. And so then we take this network and using the same broad data, we analyze this and find that basically repeat strategies are very common in this space. New strategies are very uncommon. It's enormously stable, essentially. Once the data has initiated the models, which is in the mid-1980s, then really these are enormously stable. The challenge with that is that there's an explosion of new opportunities. Right? There's an explosion of new chemicals, new biochemical opportunities to bridge and to introduce new elements into this space, which suggests in our model here below that people have to prefer essentially focusing within their fields over identifying this new space of opportunity. So it's not just that people are wandering through this space of opportunities randomly, that they're essentially constrained and focusing within existing subfields. It turns out that people do get rewarded for taking riskier strategies, but that reward doesn't fully compensate because if you engage in something like a jump, introducing a new element to the scientific space, then it's much more likely for you to fail. And in fact, we find that scientific investments themselves aren't rational. If they were rational, then and scientists were just trying to maximize citations, then they would be much more likely to take the easy out, basically. And the reason being here is that there are some things that just shouldn't be combined. Like there are certain strategies which are fundamentally risky. You're combining things that just they shouldn't be combined. They should be uncombined. So what motivates this risk-taking? Well, we look at exceptional achievement and prizes. For example, we find that these high-risk strategies end up being much more loaded on the papers that are associated with really outsized citations, elite prizes, et cetera, in this space. I'm about 2 minutes down in the negative, but I'm going to share one last finding, and it's not even, I basically, this is a simulation model here where I want to just say, okay, now that we understand that this traces broadly what one might expect that scientists do, then what if we build a generative model, we estimate that model to see what scientists do, which is broadly what we just showed in our non-generative framework, and then we simulate a whole bunch of other possible options. So we have this relatively simple model where people just pick things as a function of their centrality and their distance in the space, and we estimate that from data, and we find that basically people pick like really important and really kind of like unimportant things that are really close to one another in time, and then we estimate this from a random, structurally similar sample of the network. And we do this many, many times and find that basically the scientific strategy that we identify as characteristic of scientific action ends up being really efficient for discovering about 15% of what it discovered and really inefficient at discovering like all the things that it discovered. There are many more efficient strategies. And why is that? Well, it turns out that as a field matures, that a riskier approach ends up, or riskier experiments end up being more profitable for science as a whole. But it's very difficult for a scientist with a fixed budget of experiments, essentially, to undertake, the 1 in 10,000 experiment, right? And so basically, science matures, then you want a very different set of budgets and set of incentives for scientists to approach the big problems, the right problems. In fact, we even see that basically people get more conservative over time when we estimate what would be the optimal at each point in time The approach in the optimal algorithm would be precisely the opposite. It would be less risk averse earlier, and it would get essentially more and more risky over time. And so, we can use this to simulate alternative regimes, like uncoordinated regimes in which failures are unpublished versus forced coordinated regimes in which all failures are published, and we show that they're about 5 times more efficient if people actually shared what they failed in. Anyway, how does science think? I think through stable, conservative, transmitted research habits. It's not sensitive particularly to the changes in this enormously growing possibility space. It's characterized by a tension between imitation and award. It's efficient for only discovery of about 10 to 15% of what we know, and it's much more efficient when failures are published. And it suggests that there are levers associated with policy, that encouraging high-risk work in ways that explicitly decouples productivity from job security, that funds people, not products, that allows them to build their own portfolios, that lowers the barriers to funding risky projects, that incentivizes the publication of failures in general, would accelerate these things. So I'll close with that. Thank you.
That's great. Thanks, James. I think for the use of time, we could do maybe one quick question, or we can hold. Does someone have a quick question? I think maybe they're still absorbing that one. So why don't we just hold it, and we'll go on to Mark's talk, and then we might have time for a conversation. Thank you, Carl.
Sure.
Oh, good. Thanks, Carl. Mm-hmm.
How do you now operate your own lab? What did you do differently after you learned all of this?
Actually, I've been chatting a lot with this group called Open Philanthropy in San Francisco, and trying to build, rich index of the world's problems that are driven by data, not just by kind of like back of the envelope cost benefit analysis, but they're really driven by streaming data on these problems. So I realized, like, I'm in this rat race, like I'm running on that spigot, and so I'm actually interested in finding rich ways to build updatable maps that sensitize myself, as well as the community to big problems.
Maybe it can be added to the science forecast. All right, well, let's thank James. Give a round of applause. Thanks. So we're going to welcome now Mark. Thank you for holding this position. Usually we give this to the person who's going to generate a lot of thought, so thank you. And Mark comes to us from the Biomedical Informatics. He's the Albert Williams Professor. And he's going to talk at Yale, and he's going to talk to us about analyzing the structure of genomic science. He's got a role with the Data Analysis Center for NIH at the xRNA Consortium, and I think this will round up. We've had a lot of interest in the intersection between these questions of science and the biology. So, sure. Thank you.
Thank you. So I'm going to talk to you today about analyzing the structure of genomic science, and I'm going to focus on the diffusion of ideas. So let's see if I can do this. So I first wanted to I'm very interested in general in text mining, literature, and looking at literature on a large scale. And I just wanted to say a little bit on the motivation for that. Now, we've heard a lot today about mapping science, and that's what I'm going to focus on. But I want to point out that a lot of people interested in the mining of literature also are doing this just to do better science. A lot of times we think we can find new connections between genes or new scientific discoveries, basically by doing large-scale mining of the structured literature. And we also have this idea that if we have a structured literature and we can mine it, we can also make science just more understandable. We can mash up science with non-textual explanations for things, with other sources of information. We can just make the literature better in general. And so I think that's a good thing. But of course, today what I'm going to focus on is the mapping of science itself through looking at the structured literature. Now I'm going to talk about sort of basically 2 things today. First, I'm going to talk about kind of looking at the publication patterns of large consortia. I'm going to focus on this one consortia, the Human Genome Annotation Consortia, the ENCODE Consortia. And then I'm going to talk a little bit about building a kind of quantitative model of information diffusion. And I'll go through these. So first I'm going to talk about this consortium and so forth. So first of all, I just want to say that as you've heard in many of the other talks, more and more science is dominated by the consortia science, group science, big science, and so forth. And we're all interested in the dynamics of that. And a number of years ago, we made a little tool that sort of sat on top of PubMed. We call it PubNet. And you put in a PubMed query, and it makes a bunch of pictures for you. It makes little networks and stuff like that you can look at. And there's many different types of networks you can make. Of course, you can link papers by sort of shared mesh terms, by shared locations. But what people often find the most intuitive is a kind of linking authors where a linkage between them represents a shared co-authorship. And so I'm going to be focusing on these types of graphs mostly today. So when we first applied this tool, this was a number of years ago, to an earlier endeavor, and this was called the Structural Genomics Endeavor. This was an NIH initiative focusing on kind of the structure of the proteome. They had a number of funded centers. They're shown by their initials here, and this just shows the author structure in these centers. And what I just think is interesting is you can see just by looking at these pictures, very different social structures, very highly centralized. This center seems to be split into a number of parts. This is a sort of more decentralized organization. And I think that's just easy to see right away. So now what I'm going to focus on is these sort of genomic centers, and the way a lot of the Genome Project has worked over the last few years is you kind of have the accumulation of large amounts of data, and then this what they call a rollout, a huge publication. Of course, the granddaddy of this was the publishing of the human genome, which was this big race. But then following the human genome, the NHGRI launched this effort to annotate the genome, and there was a number of initiatives. The center of this was the ENCODE project, which had its rollout in 2012, but they had pilot projects, the ModENCODE project, the pilot ENCODE project, and after they had the what they call comparative ENCODE project. They kind of compared the human genome to mouse, worm, and fly. There's also been very big efforts, maybe you've heard of the 1,000 Genome Project, where they've looked at human genome variation, a very large scale number of rollouts for this, and more of these rollouts are very popular because, of course, you get the cover of Science and Nature, and you have a lot of impact. I mean, that's the main thrust behind them. So I'm going to be talking about these rollout structures. Now, hold on. Let's see if I can, oops, there we go. So I'm going to be focusing on ENCODE. Now, NHGRI, in particular, the NIH, what they're really interested in is that the project has a big impact, but also people use the resource, okay? And the program staff care intensely about this, and they actually track very carefully not only the papers that ENCODE researchers publish, but they also track the papers that are published that don't involve the researchers in the project, but which use the data from the project. And they, of course, want to build a resource that's going to be useful, so they track this. And so this data is actually generated by NHGRI staff, and I'm just plotting it for you here. This is the papers published by consortium members, that's blue, and then people using the data. And the important thing, of course, that the NHGRI cares about is that these red bars really go up. But notice it takes a long time, okay? What's interesting, this is the histogram of the number of authors in those papers. What's interesting is that the ENCODE papers obviously have a lot of these big papers. These are 150 author, 200 author papers. But sort of the outside people, a lot of the people using these data sets are also big science too. So now, oops. So what I've done is I kind of used the PubNet thing, and I kind of made a network of this. And I'm going to explain how this network works. So I did a network for each year here from beginning 2004 to 2014. I colored the people whether they were ENCODE members or non-members, and I'm going to explain. I have this concept of a broker I'm going to come to, and I've kind of arranged these networks in a way to accentuate a pattern. And what you kind of see is the development of this ENCODE core, okay, with this sort of yellow core, and then this ring around it of these people using the data. And you can kind of see it graphically here. But what's interesting, of course, is when we look at the network statistics, we can actually see something kind of neat. Well, we can see, obviously, we break the graph into clusters or modules. We can see the number of modules of these data users much larger than the core users. And the network statistic called the modularity, which talks about how connected each of those things are, really becomes very strong for the consortium, which really publishes as a unit. And then sort of you can see the modularity statistic actually tracking these big rollouts, whereas this is not the case for the data users. What's also interesting is if I plot for each of the people the number of neighbors they have in the consortium versus the number of neighbors they have outside the consortium, there's a particular group that emerges, this is this broker group that I've alluded to, that have a lot of connectivity both into and out of the consortium. And these people we think are very key to mediating the information flow, or the diffusing of the data, quite literally, from inside the central group to outside the central group. These are people that are connected into both groups, and you can see quite graphically them mediating this. Now, I did this analysis also for another consortium very closely related to NICO called ModNICO. You get the same type of results. I just want to point out that we can look at information diffusion and data diffusion in another way. Another way of looking at very crass, and it's not crass way, but a very sort of gross way of looking at it is just looking at the amount of bases associated with various publications. And you probably know Nature is the preeminent genomics publication. Tremendous amount of cumulative bases, and obviously a lot of big science publications, Science, PNAS, and so forth. But what's really neat to see is these things right down here, 2014, 2015, popping up exponentially. These are like the Journal of Molecular Ecology, the Nature Chemical Biology, journals representing completely different fields which are suddenly starting to use this nucleic acid sequencing data. And you can see the diffusion of this technology through the data, the data sets being produced and used. You can also look at diffusion of an idea or a technique just by simply tracking keywords. I did this paper a number of years ago when RNAi came out and really revolutionized a lot of molecular biology. One of the really neat things about it, which wasn't appreciated by its founders, was it had a completely unique term that never occurred before. RNAi, you never saw that before. So you could actually track perfectly the development of this term in the literature, starting from 1998 when the two protagonists, Andrew Fire and Craig Mello, who later won the Nobel Prize, first started publishing together, and you could watch them publishing together and other clusters of people using this term kind of surrounding them, finally to when they kind of separated and published in independent clusters, and finally to a place where almost like you almost had a phase transition where everyone was using this technology, and everyone was essentially connected to everyone because it was such a common technology. Okay, so now I'm going to talk a little more about information diffusion and building a model of this. And I'm going to shift gears a little bit. Now I'm going to talk about using data from what's called the PLOS article level metrics data. Now, PLOS, you probably all know, is a very open science motivated thing, and they have this data set where they exhaustively track the accesses and uses of all their papers. And you can get vast amounts of statistics about each paper, PDF downloads, HTML views, comments, and so forth. And, for instance, here's HTML views and PDF downloads for typical papers, and you can see they're roughly correlated. And most of these statistics you can see in this heat map are fairly correlated, but some are not correlated, and I find that kind of interesting. For instance, XML downloads, not that correlated actually with HTML views. And now if we look at not a typical paper or the average, but we look at the distribution of papers at a particular time. So if we look at a certain number of months after publication, how many downloads we get for a thing, we'll get a distribution around this average, okay? And we find these distributions empirically have a log normal structure to them. They look like this, and so this is what that cut would look like. And the fact that they have a log normal structure to them suggests that they can be modeled in a particular way. In particular, they can be modeled by a random multiplicative process. And so we developed this very simple model. And it's a very intuitive model. It's basically the cumulative number of accesses of a paper at a given time is the number of accesses it has at a previous time, but increased by a sort of word of mouth or transmission. Then there's a sort of transmission parameter, and we break that into a a sort of paper-independent quantity that sort of modulates the drop-off of transmission of a paper to one that's dependent on the papers and dependent on this random process. And since this particular parameter, this paper-independent parameter, is independent of papers, we can refine its value. And in particular, one of the things we find is there's a kind of nice fall-off in these things. There's a very fast decay. It's sort of obvious, the papers are accessed right a lot, and then they decay, and then there's a slow power law fall-off. And you can think of how this relates to kind of the fame of a paper or the response of readers to it and so forth. Okay, so I'll just summarize kind of quickly what I've talked about today. So I've told you today about kind of looking at genomic science and mapping the diffusion of ideas and data across this discipline. And first I told you a little bit about this tool we've developed, just kind of graphically analyze the data, and you can see when you look at different consortia, you get very different structure. Then I focused in detail on the ENCODE consortium and looked at kind of its evolution. And here I think you can really see the difference in the structure between the users of the data and the consortium itself in terms of the modularity and the number of clusters and so forth. And also, this work really points to the key role for these brokers, just very simple people that really connect these two groups together. Looked at other examples of data diffusion, for instance, the RNAi example. And then we talked a little bit about developing a quantitative model of information diffusion, a very simple model based on the data from the PLOS article level metrics. And the key idea was this random multiplicative process, which you see in many different contexts. And the key idea here was there's a time parameter, and it really has two different regimes. This sort of fast fall off, and then a sort of slower die out. And you can see that fits the data fairly well. Now I'll just say who's worked on this. Most of the work on this was really done by two research scientists who work with me, Daifeng Wang and Kun-Q Yan. They developed the information flow and the analysis of ENCODE. I've also alluded to some stuff that was done quite a while ago by Sean Douglas, who was an undergrad at lab. He actually built a lot of the PubNet stuff. And a lot of these visions for text mining were from working with Andrei Rozhetsky, who you heard about in the previous talk at the University of Chicago. And with that, I'll thank you for your attention.
Questions for Mark or for Robert or James? I think kids were getting to that time, but I'll ask a quick one. I was curious, the RNAi as a term, that seemed like it was interesting. Do you know, are there other terms that might be worthwhile to run a similar search on, or has anyone looked at that?
That's exactly, I'm a hound for those unique terms. And actually, molecular biology is great for cryptic terms. I mean, CRISPR-Cas, that is a bizarre term. I'm sure people are going to be searching their way to the Nobel Prize for that. But yes, there's a few others.
So I'm just curious, just because I know that, you're looking at not just the structure of these collaborations, right, and the kind of the center and periphery of these things, but how does that shape the relative concentration on problems in those spaces, right? I mean, are they different classes of problems that kind of correlate with these clusters? What are the consequences, essentially, of this kind of political economy of access to the data and of collaboration for the scale of problems, the type of problems that people study? I mean, are there consequences?
Well, I think that's a good question. I mean, I don't know the definitive answer to that question, but I think the canonical answer to it would be that, for instance, the consortia is mostly focused on building a genomic resource. And what they really want is they want the people outside the consortia who are really working on different problems to be using that resource, in pursuit of that problem. So that would be the canonical answer. Now, I don't know how true that is. I'm saying that's the kind of belief. Yeah, that's the kind of belief that people have. I don't know if the two groups really are that different in what they're thinking about, but that would be the kind of mindset that you would bring to it.
So whenever there's a consortium and then you have a roadmap coming up, that's like a kind of external shock to the system. Have you looked at other external impacts on the system, such as new funding mechanisms or major discoveries, and how these publication and also co-authorship networks change in response to this? Because typically you really get to understand a beast, such as the one in the chamber up there, when you start to interact with it or when you throw something at it.
Well, I think that's a very good question. I mean, I haven't looked at that, but obviously the human genome rollout, I think around 2000, was a huge media blitz on that, and I think it probably had a lot of impact on future funding opportunities, but also there's a lot of reaction to it. And and you saw the same thing in 2012 with the huge annotation rollout. In fact, it was, I would have to say, a fairly negative reaction on the part of the scientific community to that. And I think that in a sense it's kind of interesting, seeing like a little science storm, seeing that reaction, counter-reaction. I think it's more, you can see it in papers, but they're much slower. I think the way to study this, of course, is like in Twitter or in, blogs and stuff. But
I think we have time maybe for one last question before we close the panel. You got one? I got one.
Sorry, this is just a great panel. So I already asked James Evans what he changed in his behavior after doing these analytics, and I would like to ask the same question to Robert and also to Mark.
So how I've changed in doing these analytics and what we've changed?
No, after you have seen how your community reacts to these changes over time, and what your role in that system actually is.
Well,
you change your behavior, how you run your lab, how you select collaborators, or how you go for funding opportunities.
Well, personally, I'm both, I guess, a participant and an observer in this, some of the stuff I'm showing, which I think does give me some insight, but also probably colors my judgment a little bit. My personal sense of the community reaction to things like this, unfortunately, is I don't know if they're that into it. I think that the particular community I'm in, I think I don't think that, I think the NHGRI and the funders are interested in this because they're intensely interested in the usage of the resource. The scientists themselves, or most of the scientists, are probably not that focused on this, which is probably the reason I can work on this without them even competing with me in the tiniest bit. I don't even notice. Yes.
I have a question. From the point of view of the funders...
Let me just mention that, to follow up with Kati, so our work is so macro that there is no easy way to answer the question, but I will report that I gave the talk recently to a bunch of investors, and the investors are very keen to know what is the sixth technological wave, and if anybody has any ideas, I know some people who would like to know the answer.
I had a question. It seemed to me from your analysis of ENCODE, I think it was, that you look at different centers that some were more centralized, some were more distributed. So it looked like an experiment in itself. Can you now say which ones were the most successful, attracted more attention, or created the most impactful papers, or whatever, some sort of metric?
You could certainly do that. I didn't choose to do that, but you could certainly easily do that. For each of those centers, you can look at the subsequent publications or any metric of success and ask if that split up center is somehow less successful than the unified one. I didn't look at that, but I'd be quite interested to know.
Okay, thanks.
Thanks. Well, I think we have one last transition. First of all, I'd like to thank the panel. It was a real pleasure. And I think we have time for some concluding comments from Masha and Kati. So I'd like to welcome them. Looks like Dan's about to set up. I guess maybe one, while we take the moment, I would like to personally thank. So if we could maybe have a round of applause for our organizers, Kati, Masha, and Daniel.